Cool. All righty, we are going to get started. Hello, everyone. Welcome. Thank you so much for coming. Thank you for everyone online. It is a pleasure of mine to introduce Dr. Dana Chadwick today. Dana is a scientist at JPL, the NASA Jet Propulsion Lab, and has had kind of a beautiful and, in my opinion, envious career. I think you've done an amazing amount of work, even at an early stage. So originally got her PhD from Stanford, has done a variety of postdoctoral positions, including was an NSF fellow at a split position between Stanford and Lawrence Berkeley National Labs. Also did some postdoc time with the Carnegie Institute. And I feel like one of the things that I'm really excited about with Dana is that we know we're living in this time where we're getting amazingly new techniques coming into the world, and I feel like Dana is right at the forefront of all of these different approaches, and yet is able to stay really grounded in true, like, basic, fundamental theoretical science. And so as somebody who is not as familiar with some of these concepts, I'm always so grateful to talk to Dana, because it feels like talking to an ecologist who can then bring in this amazing sense of work. And as you'll see from like her CV and probably today's talk, you've worked across so many different scales, and that's always so fun for me. So I know we're going to hear about a lot of cool stories today. And so with that, I'll hand it over to you. So thanks so much for being with us.
Great. Well, thank you so much. That was a very, very kind introduction. And thank you guys for being here today. Is the mic close enough? Is that working okay? Yeah. Okay. Great. Yeah, I mean, it's really great to be here. You know, I am at NASA now, and I do a lot more programmatic work, but it's really fun to come and talk about more fundamental science and kind of get back to thinking about more of the research side of things that I really do love. So I'm going to talk today about a series of studies that I've done over the years, thinking about this kind of biogeochemistry at landscape scales using remote sensing technologies. And I like to start this out by using this kind of diagram of the critical zone. This is from John Chorover and the CZOs. And it's kind of a depiction of what we'd like to understand about the critical zone, which is, of course, the top of the vegetation canopy down to unweathered bedrock. And there's just a lot of aspects of this that can be very hard to characterize. And oftentimes what we really can observe is actually looks more like this, right? The subsurface is often a black box and something that we do a lot of modeling. We do some point measurements, but it can be really hard to understand. And so one of the things that I really think is exciting is how can we leverage what we can observe to better understand the subsurface. And this idea, of course, is not new. So this is an example of, well, this is a Google Earth engine, but from soil webs. You can see that this is an area in the Colorado Plateau, and if you overlay on top of this, variable vegetation and variable topography, a soil map, what you see is that traces really well back to the vegetation and the topography. And in places like this, where you have differences that you can see really easily with your eyes, you can start to do it. And this is actually how soil maps are made, of course, right? And this is why it can be a bit circular as you think about soil maps and vegetation properties. But as you get to the humid tropics, oftentimes you have landscapes that look like this. So this is actually not a picture. This is an image from an imaging spectrometer. This is just the red, green, and blue channels, so like what you would see with your eye, overlain on a LiDAR topography map. But as you look at this, you're not able to see a whole lot of variability that's associated with either changing vegetation communities or an understanding of the topographic variability. And so what's really cool, I think, about the age that we are in right now and how we can use imaging spectroscopy data is that you're starting to be able to take those and leverage them to understand variability across the landscape that is not visible to the eye. So this is an example of a map of vegetation traits, including red is leaf mass per area, green is calcium, and blue is phosphorus. And it gives you a sense that as you move across this landscape, there's actually a lot more variability than what you can see to date. So I'm going to start out by talking a little bit about imaging spectroscopy as kind of a background and then go into some of the examples of how we can utilize these. So, oh no, is it going to auto advance? Yes, it is. I checked this, I swore. But hopefully it'll just stop once it gets to the end. It's not going to stop. Awesome. Okay, so what you see up here is a reflectance spectrum. So this is actually from the EMIT mission. So this is a sensor that's on the International Space Station right now. And what you're seeing here is they're a little faint, but you have this blue, green, and red lines. So those are the portions of the spectrum that you see with your eye. And then we have this whole area in the near infrared and the shortwave infrared that we're also able to capture. And this is at 7.5 nanometer intervals. And what you're seeing here is a pretty characteristic vegetation spectrum. But as you look across these different types of surfaces on the planet, you can see that there's a huge amount of variability in the types of reflectance characteristics that we get based on what you're interested in. So geology, we have these incredible mineral spectra that we're able to learn a lot about the distributions of different deposits, a lot of variability across ecosystems. People are doing a lot of work with snow and ice reflectance spectroscopy to think about basically the albedo and the snow-water equivalents, so kind of thinking about predicting water resources over the course of the next year for snowpacks. And then there's a lot that we can do with aquatics to think about sediment load and a variety of other characteristics, including submerged aquatic vegetation, et cetera. And it's an exciting time because we've really come a long way in the technology here. So I think I am contractually obligated to point out that the first imaging spectrometer was made at JPL AIS in 1982. And we've continued to advance. So AVARIS, the original AVARIS is huge. It's actually still in operation. It's going to be retired soon. But this was kind of the next generation of airborne. Then this is the same guy. There's some time elapsed. I like to give Michael a hard time. But so we moved on to the AVARIS next generation sensors. These are sensors that were built at JPL, but that also support the NEON program. So they're all the same generation. And now we have EMIT, which is on the space station. And we also have a series of airborne sensors that are consistent with that type of build. AVARIS 3 and AVARIS 5 is now online as well. And we're looking towards a future where we should have the Surface Biology Geology mission VSWR sensor in orbit, and that will be not on the space station anymore, but a free-flying satellite with about a 16-day repeat, 30-meter resolution. So that's a Landsat-like type of observation where we'll be able to really start to leverage these data kind of in a very consistent way. So it's a really exciting time to be thinking about how can we leverage these and building these case studies and understanding of how are you going to use them as your careers progress. So I'm going to start and talk about a variety of different studies, and all the ones that I'm going to be talking about today are airborne. I personally love airborne. I think it gives you a really amazing perspective at the scale at which you and I actually operate. A 60-meter resolution is great for a lot of things, but 1 to 2 meter resolution really gives you really awesome insight into ecosystems. So I'm going to start out in the lowland Peruvian Amazon, and there we're going to have kind of a fairly consistent, I kind of think about the state factors concept here, and so kind of consistent potential biota, consistent climate, but we've got some topographic variability that occurs in there, and we're going to be able to kind of think about how that impacts nutrient cycling and how we can see that in the vegetation characteristics. We're then going to go to Malaysian Borneo and think about this very large elevation gradient with a lot of very different lithologies. What can we learn there? And then I'm going to talk a little bit about work that was done in Colorado as part of my postdoc, but also was reflown again this summer. So there's some interesting kind of time lag activities that we can look at there. And then focus on a project in California, which was the SHIFT project that I led the ground campaign for in 2021, and this was focused on kind of weekly observations, so this really sub-seasonal dynamics and what we can learn from that. So I'm going to do a brief overview of the airborne component of this. So we're going to be looking at both LiDAR, which may be a little bit more familiar to folks. So it's an active sensor, of course. And so you get these images of vegetation structure, but I'm primarily going to be using them to remove the vegetation and think about the surface processes and understanding how we can think about erosional dynamics when you are able to see through the vegetation canopy. And then we have a V-SWIR imaging spectrometer. That is a passive sensor, so we're using the sun as our light source, and that's where we're getting this type of reflectance spectra from. So currently, we are still at the phase where we're generally having to, every time we want to use imaging spectroscopy to think about vegetation properties, we are retraining these models for different systems. So I'm going to kind of go through the process of how that works. And then I'm not going to talk about it for every study here, but know that this is a process that really is very labor intensive still, and we are trying to get away from that, which is kind of one of my big efforts currently. So this is the example from Peru, and what you're seeing here is all of the samples, all of the places where we sampled for vegetation properties. So in red are those canopies. That's not to scale. You would hardly be able to see them if they were to scale. And of course, this clearly follows a trail network, right? So the access to these, you're climbing the trees, you're trying to actually sample from the top of canopy vegetation so that you're capturing the same thing that the plane is seeing as it flies over. And you're trying to do that within a pretty close window of time so that you don't have phenological change or other types of changes that would alter the chemical composition of the samples that you're taking. So you have this combination of the physical sample, the geolocation of where these are, and a delineation of the crowns from that imagery, and then you're able to actually extract the reflectance spectra associated with each of those crowns. And when you do that, you can develop these models. Generally, people use an iterative partial least squares regression style model. People are playing with all the different cool AI, everything in the world. This has still served pretty much as good as anything else to date, though it would be great if people have other strategies. From that, you actually get a linear set of coefficients that you can reapply to that spectra, which makes it really efficient computationally. And then you can come back to your image, which each of these pixels has that whole spectrum in it, and you can apply a model. For example, this is a nitrogen model, and you can start to see that you have this highly variable nitrogen concentrations across this landscape that you're not able to see. And I always like to point out, especially to folks in this room, this is, if we're assumed that in the tropics, this is about a 30-meter diameter crown that we're looking at, this is 50,000 tree canopies that we're looking at in this one image. And that's obviously, you can't even think about climbing 50,000 trees, right? I can't. I'm not in shape. And so, this is just really kind of a step change in what we can understand. And so this is an example, again, from this site in Peru where we're looking at differences across this area. And just to kind of give you a little context, this is where two rivers are meeting. And we have some characteristics that you can see here. So in this is an old landing strip. And this is some old areas associated with mining activities. And these are areas where there are bamboo is regrowing. And we do have samples of bamboo that indicate that they do have very high nitrogen concentrations, but no bamboo samples were actually used in the generation of these original models. So it's a nice indication that we really are doing a decent job here. You also see these low leaf mass per area values at these early successional areas associated with meanders. So again, that's what we would expect from ecological theory. Oops. And then the one other thing I wanted to point out is that we have this area here. So these are old terraces. And then you have the transition to these very active floodplains. And you can see that the rock-derived nutrient change there is very different and really driven by this riverine deposition that is kind of continuously increasing the rock-derived nutrients. So in addition to that kind of binary terrace and floodplain dynamics, I'm going to kind of then take a look at a little more in depth to think about, what other erosional dynamics are happening in this type of system. So this is a picture of this system that I took from a canopy, what are they called? Thing you stand on above the canopy, you know what I mean. That's just to give you a sense of what it looks like. And this site is actually really interesting because it is in the lowland Peruvian Amazon, but it is right at the bottom, at this bottom edge of the Fitzgerald Arch. So in this area, the Nazca plate is subducting under the South American plate here. And so you get this high area here. And it's a little bit hard to tell, I apologize, the screen is amazing and huge. But the rivers are actually all kind of going away from this area of local uplift that's right here. And so you have this increased erosion pressure because you have a local uplift inside the basin. And as you look at this area here, this river is kind of going up onto that arch. And you can kind of tell because of the erosional patterns that you see here, as opposed to down here, where this is like kind of more recent deposition. And this is an older set of terraces that's being eroded into. So we're going to think about the small catchments that are eroding basically into that terrace system. And what we did was to actually take a whole series of slopes associated with these and take soil samples along those hill slopes in addition to this airborne data that we have to try to understand what the nutrient dynamics are that are going on here. And so we can start to hypothesize, like, what are the potential mechanisms that might drive biogeochemical patterns across these terrace landscapes? And so the way we started to think about this was to think about incision depth. So thinking about that terrace surface as kind of the old original surface, and then basically going down into that terrace. And so you'll see a whole bunch of figures that are by incision depth. So it's not elevation, it's starting at the terrace surface and then going down. So there's a series of different things that might be going on here. And one of them is kind of in this erosion category, what's going on kind of in the geomorphology. And this is likely to impact soils at the subsurface where they're slightly less impacted by very active cycling. So I've blacked this out here. And there's a lot of hill slope erosion that's happening here. There's a huge amount of rainfall, almost 3 meters per year. And so because of the local uplift, you have streams incising down into these hill slopes. So as things are eroding off, because of this stream incision, you actually get everything that removed away from the base of these hill slopes. You don't have a lot of colluvial accumulation. You have it actually washing out to the main larger stream networks, which could potentially cause the advection of less weathered sediments into the soil profile as you have that erosion occurring. Then we can also think about the fact that this is a really wet environment, and so there's likely to be chemical weathering and leaching from the surface, so this is likely to be very depleted, kind of consistent with our understanding of the tropics. But then that can still be causing redistribution, which might lead to higher nutrient concentrations downslope. So there's a couple of different things that could be going on here, and several of them would lead to higher nutrient concentrations in low slope positions here. We can also then take the imaging spectroscopy data and think about the fact that that would give us an understanding of the vegetation's experience, effectively, of nutrient availability. And so to give you a sense of what these maps look like, this is foliar leaf mass per area, calcium and phosphorus. And what you can just visually see is that as you move into these lower slope positions, we do have an increased amount of phosphorus and calcium and a decrease in leaf mass per area, which is kind of often a trade-off that exists. So we can turn this into a graph, right, which might be a little more intuitive. So this is looking at every catchment that we were focused on here. And again, this is going to be your incision depth. So this is starting at the top of that terrace and then going down into kind of these small stream networks. And so as you saw kind of visually before, you have this decrease in leaf mass per area. We have an increase in nitrogen concentration. Leaf mass per area and nitrogen often trade off against each other, so this is what we might expect. But I do want to highlight the fact that having this landscape scale view is really valuable, right? If you looked just at this catchment that's now in blue versus this catchment that's in purple, you might draw very different conclusions about what's happening in this system. And so being able to have a perspective that takes into account many catchments gives you kind of an average sense of what's going on within an acknowledgement that there's a lot of stochasticity associated with biotic processes. So we can then look at our rock-derived nutrients and see that these are also increasing. So this is great, kind of what we would expect. But so we can also then start to think about, okay, how does this relate to what we understand about the leaf economic spectrum? And so what I decided to do was to hold LMA constant and basically think about the residuals compared to the relationship with LMA. So I'm going to keep this here. And when you look at nitrogen, what you see is as you move along these hill slopes, nitrogen basically trades off exactly with leaf mass per area. And this, so there, this is, there's no additional change to nitrogen once you take into account leaf mass per area. When you look at phosphorus, there's a slight increase as you move down these hill slopes, but it's not, I mean, it's statistically significant. Is it biologically significant? I'm not really sure. But as you look at some of these other rock-derived nutrients, you have substantial changes in addition to what you see, what you might expect based on the relationship that we see across the entire landscape between them and leaf mass per area. So I think this is kind of an interesting indication that there is likely some additional rock-derived nutrients, especially these cations, as you move downslope, which can potentially relieve some limiting pressure. So then we can return to the soil surveys and think about, okay, what can we learn from that, and can that give us additional insight into which of these processes might be occurring? So we can first start to think about, what can we learn from the subsurface soils and how that might tell us about what's happening in the erosional dynamics. So we're going to look at chemical depletion. So these are tau values for those of you that are into that kind of thing. And so if you go below 0, that means that for that element relative to a reference parent material or parent-like material, you have chemical weathering and leaching and removal of that element. And if you go above, you have some accumulation. And so what we see is that calcium is just incredibly low across this whole system. It's very, very depleted. And this is in the bulk soil. So this isn't exchangeables. This is, your total soil. As you look at some of these other rock-derived nutrients, we do see that there is kind of a reversion closer to the parent material, which does suggest that as you have this erosion and incision of streams, that you are bringing up fresher material into the soil profile. So then we can think about what this is doing to plant available nutrients. So this is just looking at a 1 meter profile, a kind of aggregated available nutrients for these different cations plus phosphorus. And we'll see that there are increases here in potassium, magnesium, calcium. There's water in here somewhere. Okay. Phosphorus is a mess. It's always a mess. This is based on dark oxalate extractions. There's a lot of different types of extractions you can do for phosphorus, a lot of different ways you can consider it. If I had more money and time, I would probably have done a variety of different types of phosphorus extractions to think about the dynamics there, but this is where we ended up. And then the last kind of way that I was interested in looking at these soil samples was to think about what, as we have this available profile information, what proportion of that is held in the top 10 centimeters? So kind of what is uplift and retained strongly within the surface soils. And so because we're looking at the top 10 centimeters, if we had a uniform distribution through the whole profile, you would expect, basically a retention of 0.1 in the surface if it was totally even. And if you have less than that, then you'd have depletion at the surface. And if you have more than that, then it's kind of biotically retained in those surface soils. So when we look across these, what we can see is that all of these are retained in the surface, which is what we would expect. These are essential nutrients. Interestingly, I want to point out just a couple things. One is that as you have this increase here in potassium, you have a decrease actually in the retention, which I think is really interesting. It's less likely to be limiting as you move down this hill slope because you are actually doing less to retain it, especially when paired with things like calcium and magnesium, where there isn't really that change as you move along these hill slopes. So I think that's interesting. Calcium is retained really strongly, and I always like to show aluminum also, aluminum not being an essential nutrient, and it is kind of not retained preferentially in the surface soil. So just to be clear that it's not, everything isn't high in the surface. There is actually, some sense here. So we have this kind of couple lines of evidence suggesting that there's rejuvenation is occurring, that we do have this redistribution and transport of calcium and other nutrients downslope, and then this strong retention of biotic uplift of these nutrients. And kind of when you put all of this together, you have this really interesting picture that, at least for me, makes me think, if I were going to do a nutrient addition experiment here to understand limitation, I'd really be focused on calcium and magnesium in this system. I think that there's a lot of lines of evidence, and then I would kind of continue to pursue those down a variety of different other avenues. But I think that it's really nice to be able to kind of take all of these lines of evidence, bring them to bear, and try to understand, how this landscape is functioning. And there's actually been some really interesting work by a student in Josh West's group at USC who was taking stream samples in several of these same catchments, and we're seeing dynamics that were very consistent with these conclusions and understanding that there's less loss of calcium and magnesium from the system relative to other nutrients. And so it's very fun to kind of see all these different techniques come to bear. So this was an example, that has, it's a similar lithology, similar climate. There are some topographic variations, but not like a large elevation gradient. So we're going to go from there to Mount Kinabalu in Malaysian Borneo, where we have not only this kind of local topographic variation, but we also have changes in parent material. There's ultramafic outcrops all up and down this mountain, and then also this very large elevation gradient that occurs. So there's a lot more dynamics that come to play in this system. And so I put this up here to remind you that we had to do all this again in Borneo to generate foliar trait maps that we could leverage. And then what I did was to think about, okay, can we look at a variety of different catchments across this elevation gradient, across these different parent material types, to try to understand kind of what's going on at the kind of these small watershed scales. So we started out by just looking at what were the canopy level averages for these traits in these different catchments. And this is good. This is kind of consistent with what we would expect with large elevation gradients in the tropics. We have a increase in leaf mass per area as you get up to higher elevations where it's cooler. You have a kind of corresponding decrease in foliar nitrogen. You have decreases in foliar phosphorus, which is, again, this is consistent from Kitayama's work. And then an interesting difference here where, for ultramafic soils, which are in orange, I didn't say that, you have inverted calcium to magnesium ratios, and in that case, you don't actually see a whole lot of change with calcium, whereas you do in the sedimentary outcrops. So this is nice to see and kind of consistent, again, with what people tend to find. But if you look at the variability within these, it's huge, right? And it can be a lot of elevation variability as well. And so if we want to understand kind of what's happening inside these different catchments and how that's playing a role in distributing nutrients, then we might want to take a little bit of a closer look. So to start doing that, there's a lot of variability here, and so I was interested in looking at kind of what is basically the rate of change in canopy nutrients across these hill slopes to kind of try to normalize this across these different catchments. And so again, gravity works. Nutrients are moving downslope. You have Nitrogen increases as you go down slope, same with calcium, same with phosphorus, and then leaf mass per area kind of doing the opposite. But again, there can be a lot of variability, and can we do better than this amount of variability that we're seeing in these bar charts? So I kind of took a step back and did do some soil sampling. It was nowhere near sufficient to say anything because these are incredibly steep landscapes and very hard to work in. And so we went back and thought instead about geomorphic theory. So this is work based on work by Josh Roaring, and who has thought a lot about ways of kind of characterizing equilibrium and disequilibrium in landscapes. So what you see here is that the idea that you have so you have hill slope curvature, you have the total relief of the hill slope, and then you have the hill slope length. And a dimensionless erosion rate can be kind of associated with the curvature and the length. So how steep is this? And this is kind of intuitive if you just don't think about any of this and think about, okay, in really, really steep eroding environments, you get these really strong ridges, right, like peaks. And then places where you have kind of lower slopes that are nice and gentle, those aren't eroding nearly as fast. So that's just kind of, they're basically working to quantify that here. And this is kind of basically the line at which you have equilibrium. So where that whole thing is in steady state. So you retain basically the same hill slope structure and the erosion is occurring evenly across the entire area. And you could have that occurring at a relatively low erosion rate, such as here, and this is kind of what I'm talking about with these broad hill slopes. Or it could be here, where you have a much higher erosion rate and you have this much kind of steeper, sharper curvature. And so that's, landscapes tend towards steady state. So they're all kind of adapting to any changes that they've had in erosion rate to get to this steady state. And again, this is some examples from mostly California and the West Coast, but looking at the fact that hill slopes on average tend to fall along this kind of steady state line. And Kinabalu, if you take all of the hill slopes that we can kind of measure this way, falls in steady state on average, but across the whole area. However, if you look at the individual catchments that we're working on, it's highly variable. And so what does it mean when we're out of equilibrium? Well, if we come back to these two, what it means is that you're somewhere in this middle. And there was a really nice paper that was done on the Dragon's Back in California by Hurst, and they basically looked at this process and showed that there's really a hysteresis associated with this. So if you have an occurrence where you're increasing your erosion rate. So we're saying that now these hill slopes are connected and there is basically a nick point that is propagating up this stream. So you have, in this area, you're starting to move off of this equilibrium. And you're doing that because your curvature at the top of your hill slope is still the same because it doesn't yet know that it's eroding more quickly. But you're starting to have changes in your overall relief and hill slope length because that is starting to respond here. So you're up kind of over your equilibrium line. And then as you get here, this is starting to propagate basically up the hill slope until you get to the point where now your curvature is aware that it's eroding faster, and so you have this. I mean, I'm anthropomorphizing, obviously, but so then you come back down into this new equilibrium. And this goes back the other way where you have hill slope decay when you have a slowing. And so they did it really is a very cool paper where you can see because of the movement of this ridge under a set of hill slopes, you can really see that playing out. So what I was interested in doing here was to think about, okay, can we put hill slope disequilibrium on this axis here? So if you're below that equilibrium line, now I'm going to say that you're in decay, and this is basically the distance off that line that you are. And then if you're here in the positive direction, you're steepening. So your hill slope is in the process of getting steeper. And you can imagine that there might be two different responses of your nutrient distribution. You could have it be going up as you move across here, and that would be the case where, okay, as you're steepening, it's similar to the process that was occurring in Peru, right, where you're advecting new nutrients into your soil profile, making them more available. You could also imagine it going the other way, where when you're decaying, you're developing more colluvial matter at the bottom, which may be more organic rich, and so you might actually have increased amounts of nutrient availability at those low slope positions in that case. And when you are incising more rapidly, you could be removing that, especially if it's happening fast enough where you're not then weathering those nutrients out of your parent material into plant available form fast enough. So there's kind of some variety of things that could be going on. And so this is then what we actually ended up seeing, which is that you do see an increased rate of change. So there's more difference in the phosphorus concentration between the ridge and the valley position when you have this hill slope steepening happening. So again, this is some really interesting lines of evidence which I would then, again, with money and time, I would go and design a series of studies where I would look at stream discharge. I would think about some more strategic sampling focused on kind of the endpoints of these examples. I would go out and think about where nick points might be occurring. There's a lot of things that you could then do to explore within these landscapes. However, instead, I got a different job. So we have this example where we're looking at soils more directly along hill slopes. We then move to kind of this geomorphic theory type framework that kind of allows us to think about larger landscapes that are potentially more complex. And so now I'm going to talk quickly about these other two flight campaigns and kind of the directions that I'm excited about going, that a lot of postdocs and students involved are kind of taking these, and then kind of tell you just briefly about what I hope my hopeful vision for the future is, and then I'd love to take any questions. So the first one was in the East River in Colorado, and so this one was an example of a massively complicated ecosystem. So in addition to this high topographic variability generally across this mountain, a lot of differences in parent material, we also have this incredibly patchy vegetation, which makes it even more complicated. So we sampled across over 400 sites, and I apologize, this isn't suited for this amazing screen, but we sampled not only the canopy vegetation to do trait modeling, but we also did a large amount of other sampling associated with kind of understanding soil composition, microbial composition, and fine root properties. And the goal here, well, part of the goal, was to try to understand, can we leverage these surface observations to extend characterization below ground better? So let's say we took that, I went very fast there, but this is imagery. This is from the NEON AOP platform. And this is just a PCA of this variability across this hill slope. And conceptually, what we're trying to do is say, okay, we're sampling in some locations, and we're getting some subsurface information in those locations. If we can build a strong enough relationship between this subsurface and what we're observing at the surface, then maybe we can start to get a better handle on what's happening in this subsurface. So we did get a Signals in the Soils program grant to look at this. I would say that a lot of this is dragging on and still ongoing, but we worked with folks at the USGS, Corey Lawrence, to think about organic carbon distribution and how we can kind of understand that distribution with we actually haven't gotten to this part yet, but what we'd like to do is understand the additional information that we can get from the imaging spectroscopy data, and can that significantly improve our predictions of subsurface carbon. Rock-derived nutrients, thinking about how hill slope recycling of rock-derived nutrients and weathering might be occurring in this, post-glaciated environment. And then trace metals. So that's something where actually Katie Grant has made a ton of progress. She just defended. And this is an example of a map of metal concentration, so zinc concentration. There's a lot of old legacy mining activity in this region, and she's actually been able to develop maps of different types of, so this is an example right here, where you have this iron gossan and mine all right in this one area, and you're seeing really high concentration of metals in the vegetation that's surrounding this area. And so she's been thinking about how can we leverage these data and information about increased metal concentrations to understand where metals are actually getting mobilized in the environment. There's a lot of toxicity and issues associated with the water supply for the town of Crested Butte. And part of the challenge they have is that the mobilization of those metals is very distributed across the landscape, and it's happening in lots of different places within stream networks. And so if we could utilize an understanding of where the vegetation is being impacted by high metal concentrations, that might help you to identify locations where you would kind of prioritize your remediation efforts. So that's ongoing. We're also trying to think about how we link these trait maps into ecosystem models. So this is work by the Lawrence Berkeley Lab Watershed Function Scientific Focus Area, if I got all that abbreviation correctly. And they do a lot of hill slope level modeling and of kind of nitrogen flux and carbon flux through the system. And they also they're also thinking about, can they use these data to better estimate kind of microbial functional groupings across the landscape. So that's work that's ongoing. Really cool work by Ben Blonder and a bunch of his lab looking at aspen distributions. So this is just a worldview RGB. And they were able to map out ploidy levels of aspen. So this is diploid and triploid levels. And they're starting the process of thinking about if they can actually then map out the extent of individual clones across the landscape. So this has been a really cool project that we've continued to kind of work together on. So, all right, so that's another one. And then finally, the last one I'm going to talk about is this California project, which was quite recent and I think really exciting. And it's really thinking about, as we move towards orbital sensors that are going to have this repeat, around 16-day repeat, what are we going to be able to learn with that? And also, what are the limitations of our current algorithms? Because every time we develop these algorithms, we tend to try to do that around peak greenness time. However, most of the world is not in peak greenness most of the time. And so what does that mean for our ability to leverage these algorithms and think about what they mean through changing seasons? So this was, we captured this area in Santa Barbara County once a week for 13 weeks. And this is just an example of kind of indices of, this isn't the fully developed trait models, but indices of leaf mass per area, leaf water content in green rather than blue, nitrogen in blue, and then just kind of NDVI as like a reference case. And just, this is a GIF or GIF, depending on what side of that battle you're on, of this over that 13-week period, and just to kind of give you a sense of how much spatial variability is occurring in this coastal system. And so there's a lot more work to be done in this. All these data are open. If you're interested in working with them, we would love it if you did. So just feel free to let me know, or don't, and just access them. So that's kind of, so these are my airborne pet projects. I love these projects. There's a lot of very cool stuff. These are such different systems, and we can learn really interesting and different things about all of them. We are, though, entering this age where we're able to start scaling this to spaceborne. So again, contractually obligated. And the EMIT mission is on the space station right now. I'm the application lead for that mission, and I love it, and it's super exciting. We have an absolutely incredible amount of data. So if you're interested in these data for places where there aren't airborne data and you're willing and able to work at 60-meter resolution, this is really an incredible wealth of information. So we are on the space station. We're right here. This is kind of, I mean, this is old now, but we have samples over like most of the land area of the planet now, and that's expanding every day. We have a ton of resources to get you started using this, and if you ever have any questions about that, I am happy to answer them. And just as a reminder of all the different types of areas that we're able to kind of start thinking about, I just think it's so cool. It's a tool that you can use for so many different types of applications and questions, and I personally love landscape scale biogeochemistry, but if you're interested in any of these other things, we're super happy to kind of think about how we can expand the use of these data, and it really is an exciting time. So I just think that, to close, I just think that the airborne data and the spaceborne data, it's all really taking us towards this new era where we're going to be able to utilize these data to better understand our Earth system and the critical zone. And I just really look forward to it. And so I'm an evangelist, and I'm happy to talk to you about any way that you might be interested in using these data. So with that, thank you guys so much for having me. I really, really appreciate it. It's really lovely to be here with you all, and that's it.
Great, thank you so much. Man, I've just learned a lot. So we're happy to take any questions from the audience first, and then we just ask that you repeat the question.
I will endeavor. Yes.
That was a great talk, and I was thinking, or I want to ask if you consider any gene expression basically related to how traits might change with the gene expression, leaf gene expression, and how that also will reflect changes in reflectance when you collected your data.
Okay, so the question was if we consider any changes in leaf gene expression and how that might influence the spectral data we're retrieving. Is that right?
And therefore, your traits and therefore your reflectance.
Yeah, so the traits and therefore the reflectance. Personally, no. I think that there is a lot of space for that, though. I think there are people who are starting to think about, the impact of disease on traits and how that is expressing. I think that some of the work related to ploidy and it's not so much the actual expression of those genes, but kind of the potential gene pools that people are looking at. But I think that that's a space that there's a lot of room for development. So I would say not specifically. I certainly haven't specifically. Yeah.
Cool talk. You talked about the process of taking one of these the reflected spectrum, and sort of calibrating it to random samples of chemistry, and about needing to do that again when you get a different landscape. How much carryover is there? Like, does a spectrum for foliage that's rich in phosphorus or whatever here look kind of like the spectrum for foliage rich in that element somewhere else, or are they totally different?
Yeah, so the question was, in these cases where you have to recalibrate the models again and again, how much consistency is there between these different ecosystems for things like different nutrient concentrations, and how much consistency in the reflectance variability is there? Is that?
Yeah.
Yeah. Okay.
Does the spectrum of a plant that's rich in phosphorus here look like the spectrum of a plant that's rich in phosphorus over there?
Yeah. It depends. So the other thing, there's a lot of things can impact this, especially at the airborne level, because you have a lot of leaf structural information. And so, like, for example, in the work in Colorado, we did totally separate models for conifers versus broadleaf, because the needle leaf structure is so different and the light interacts with that so differently that we actually needed to separate those. But the kind of broader level, your question, we are currently, I'm leading a project we're calling VSWR Plants, which is actually trying to aggregate all of these different projects and think about how we can start to do a better job of answering that exact question. Part of the challenges that we've had over the years is that, so I didn't talk about it here, but the information that you get at the sensor has a lot of atmosphere in it. And so you actually have to basically model out the atmosphere, as we call atmospheric correction, to get a reflectance estimate. So it's a modeled surface reflectance. And different groups use different versions of that modeling process, which actually makes it so that you cannot directly compare them, which is always exciting. And a lot of that is aligned with funding agencies. So right now, NEON uses a different method than NASA does, and this is something that I'm actually work with the NEON folks really closely, and we're working on starting to integrate that so we're all using the same strategy. So at least within the US, we would have very consistent data sets. And with the database that we're trying to develop to help with this problem more generally, we actually want to go back to the radiance data so that we can take all these data that we have, starting at the radiance data, we can process it all the way through and match those with the in situ data so that we can more consistently answer that type of question across systems. So I would say that in general, if you have like Colorado and California and you have the same atmospheric correction and you have this, similar plant, maybe families, maybe even, genus, then yes, I think you have generally similar transitions between like healthy vegetation that have high nutrient status and stressed vegetation, but there's a lot of nuance there.
The atmosphere is not just that you're seeing sort of a pure reflectance spectrum of a particular element. It's like how does the concentration of that element influence the characteristics of the leaf?
Yes, yeah, and so the question was, it's not just about the atmospheric correction, it's also about how the reflectance, how the nutrient addition kind of manifests in the reflectance signature. And yes, because this isn't like an XRF, right? That's how you would like truly measure, or an ICP, destructive sampling. So we are basically looking at this through kind of the constellation of biochemistry that is enabled by enough of these different nutrients, and then the biochemical response associated with not enough of those nutrients. And so that's really one of the things that's driving this. And I don't really have a lot of time, but one of my pet things that I would love to look into is that we actually see that in systems that have kind of low concentrations of certain nutrients, that those nutrients are much better predicted using imaging spectroscopy. When you have a surplus, they're often less, especially for cations. I think that's likely because they're more easy to uptake, they're more stored in vacuoles, so they're less, like their concentration when you actually do a digest of them is less coupled to the biochemistry associated with it, as opposed to when you have low concentrations of those, it's, you are likely to have less just kind of vacuole storage of those. And I think there's a couple different cool things there. One, it would be really cool to know if that's actually true, so I would love to do some experiments around that. But the other thing that would be really cool if that was the case is that then you could use the predictive power of that as an indicator of limitation in those systems, right? Like there would be like some really cool potential feedbacks there. So a lot of work to be done. Yeah.
I have a follow-up question to that. So actually two questions. So one, how much sampling, what is the space? I'm just curious about scaling up. So if you're doing a field campaign in an area, how far do you feel is reasonable to scale across the landscape? And then how far away are we from just being able to fly over the Amazon and predicting and seeing changes without doing the field campaigns?
Yeah. So the question was, currently, how far do I feel like we can extrapolate these models, and then how far are we from having them be generalizable? So I tend to feel like generally within a region where you have a similar set of species, so I think if you were to extrapolate from the shift domain in California, north or south along the coast, and then inland, I think you have an area there where, as long as you're not transitioning to cacti, you can go for a ways there. And similarly, and this is actually a question that we can start to ask with and I think will, so I forgot to mention for the Colorado site, they reflew that site this year. So we flew it in 2018, they reflew that site, and then they reflew 2 other adjacent sites. And one of the things that we're gonna be testing there is transferability through time of those original algorithms, as well as transferability through space to these new sites where they have in situ samples from this later time. So I think we're at the, and then also they had a whole series of flights in South Africa recently, which is another Mediterranean climate like California. And so one of the projects that's ongoing right now from that is actually kind of a cross comparison of how well from these totally different species assemblages in a Mediterranean type ecosystem, how well do those cross? And I don't have the answers to that yet, but we have the data to actually start doing it. So, and then how far away are we generally? I mean, I think that it's, you know, I think that we're at the point now where this is why this is kind of my pet thing that I really want to move forward is like, I would be really nice to be able to start delivering the community with a consistent set of products from these types of measurements. So like consistently deliver canopy nitrogen estimates so that the community can also help stress test this. And so that as we have new airborne campaigns that people can start to add their data into these databases to make this more representative of different areas across the globe. That I think that this is something where we're going to continue to want to add kind of new and different, you know, examples of species or examples of structures and, you know, plants with very different leaf area structures, this type of thing, that as we continue to build that database, we're going to have a much better picture of that. So I'd say that we are not there yet, but I do think that we have, once we can aggregate them together into something that is interoperable, we have a lot of information where we could start to be answering those questions. So. Yes.
Really cool data. I had a question that kind of related to those, and I guess tying in also to your comment about like the concentrations affecting the predictability.
Mm-hmm.
I was curious, are there specific elements we have a good idea should be better predictable within distribution, like within a landscape, or potentially across distributions, just because their spectral signatures are more unique than other elements? Or I guess are there like lower hanging fruit, sort of like the elemental toolkit?
Yeah, so I guess I would say that most of the things that are lower hanging fruit would not be the elemental composition. So like then phenolics, things that have like pretty clear absorption features in this spectral range, right? I mean, you know, chlorophylls, like things that, pigments, things that actually like are specifically themselves interacting with light in this region of the spectrum are definitely the, you know, more low-hanging fruit. Like water, for example, leaf water content is very well predicted and very consistently predicted across ecosystems. And that's because there's actually like water absorption features that occur that are very obvious. And, you know, so it's like, yes, it makes sense that you can do that consistently. So the elemental piece is much more, you know, kind of transitive property via the biochemistry, which makes it harder because, you know, a lot of times we don't, I mean, I certainly don't. I feel like we need a lot more work in like the molecular, like plant molecular biology realm to better understand some of the pathways and, you know, areas where those are used across, you know, the huge diversity of plants on the planet. So, yeah.
One question here.
Very nice.
So I'm always kind of curious about the availability of some of these nutrients, and I think, you know, the assumption is if it's in the leaf, it was available to the plant to get at some point. But like, how frequently do you feel that there's nice tracking between soil nutrient content and the foliar content, or are there often times where it's actually really enriched but not so much in the plant? Or yeah, like how are you, is it just as good as you have done your own analysis for like how available some of these nutrients are, or how often do they track each other?
Yeah, I mean, we can kind of even go back and like look at some of these things. I mean, I think that there's always a challenge here, right? Because these are like a snapshot in time, and both from the airborne and from the soil, you know, measurements. And so it's kind of, I think, like a good indication and the best we can do, but like what, I feel like what you would want to do would be to kind of think about, this is a mostly evergreen forest, but some things do shed their leaves intermittently, especially if you get a particularly dry period. But there's leaf out, there's phenological variability, like there's all kinds of things that you might want to sample leaves as they're leafing out to understand, like, how much are they changing, and are they retaining this, and then are they, is there retranslocation happening? Like, yeah, I feel like, a lot of this is trying to assemble enough evidence across, and I think that's why the stream data, I think, is super interesting too, because I think that tells you something about what's leaking out of the system. So, yeah, I don't have a great answer for that. I think it's hard, so yeah.
All right, we are at 12 o'clock, so let's thank Dana one more time. And thank you everyone online and in person for coming today.